Welcome back to the Marius method. Today we're going to be talking about the cell membrane, also known as the plasma membrane. We're going to be touching upon the fluid mosaic model and the cell wall and the plant cells. We're really going to be breaking down the mosaic model as it's very complicated, a lot of colors, very artsy-fartsy. We're going to break it down. Let's get into it. So let's take a look at this diagram that we have here. You're going to see my little face in the corner. How's it going? So when we're breaking down the fluid mosaic model, let's break this term up first. Fluid mosaic. So when we think of mosaic, it's made up of a lot of different things. It's made up of proteins, it's made up of different molecules, right? And fluid, it's constantly moving. If you watch my fun facts, you know that it's not a solid structure. These phospholipids, these little heads, we're gonna zoom in here as you can see, those little heads are constantly moving. They're fluctuating to bend and form to the different things coming in and out. So what's important to know is in this fluid mosaic model, it's fluid. If it is not moving, that means that nothing can get through. So there needs to be a constant fluctuation of motion for things to pass through the membrane. Now, what I want to get into before I talk about these phospholipids, before I talk about these protein channels, is the cholesterol. The cholesterol is a key part of the fluid mosaic model. People hear cholesterol, I have high cholesterol, whatever, I'm having heart problems, whatever. Cholesterol is not a bad thing. Cholesterol is filled in the membrane. So we're going to zoom in here. The cholesterol connects the phospholipids together. And we're going to get into what those phospholipids are in a moment, but it keeps the phospholipids from drifting apart too quickly. So when something is constantly in flux, obviously there can be separation, and it happens under certain temperatures. So when it gets hot or temperatures increasing, what's going to happen is these little phospholipids are holding it together and stopping it from in the heat expanding, okay? And the other role is it keeps them apart, so when it gets cold, they don't stiff. They don't get too tight and get too close together. So as the cell membrane gets too cold, cholesterol is keeping them apart, okay? And when it gets too hot, cholesterol is keeping them together. So that's an important thing to remember. It's cold, keeping them apart, and then it's hot, it's keeping them close together. So when the heat, it's expanding, the cold, it's tightening up. The same thing happens with your muscles, right? You're out maybe on a run, you're out playing basketball in the cold. You'll feel your muscles get tighter in the cold, and then when it's hot, you may feel looser, it's easier to warm up and get in the groove. I want you to think of that analogy when we think of the cell membrane. It's a good way to remember what that cholesterol does. The next thing we're going to get into before those phospholipids, we're going to talk about glycoproteins and glycolipids. And I want to first focus on those glycoproteins. We're going to zoom in here. As you can see, the glycoproteins are on the outside of the cell membrane. On the outside. So when you see something on the outside, it's obviously has something to do with stuff coming in. So it is a major part in extracellular signaling. So I'm going to try to circle it right here. So basically, anything coming in, it almost acts as an antibody. It plays a big part of the immune response. In fact, it is an antibody. So I want you to think of, glycoprotein immune response. It is a sugar, right? It has that glyco, that prefix of glyco. Anytime you see something with glyco, it's most likely a carbohydrate, a sugar, and this glycoprotein, they are carbohydrates. They're proteins that are carbohydrates, and they have sugars attached to them. So they are located on the outside of the cell membrane, and they deal with cell recognition, so anything coming in. And similar with the glycolipids, they're typically also found on the outer surface. We're going to find one right here, and they also participate, let's zoom in, and they also participate in cell recognition, okay? And they help, maintain the structural integrity of the membrane, and they stabilize the cell. And remember, even though it's fluid, it can't be too fluid, right? Something's too fluid, it's gonna get all, too loosey-goosey there. And we're gonna need to maintain some sort of structure to an extent, and the glycolipids help with that. But they also, once again, act as signalers. They have receptors, as you can see right there. And for hormones and anything that is going to influence the cell's activity. So they're also controlling what's getting in and what's getting out, and they contribute to the flexibility, but they also contribute to the structure. So that's something to keep in mind. They do a bit of both. So now we're going to get into these phospholipids. Now, phospholipids are composed of a hydrophilic head and a hydrophobic tail, as you're seeing on the image here on the screen. But lipids, regular lipids, are going to be hydrophobic. Okay, so I'm going to try to zoom in here. So most of the phospholipid it's made up of a carbon and a hydrogen or hydrocarbon or you name it, and that's going to be half of the phospholipid. What's a phospholipid different from other lipids is that it has a charge, right? A lipid is hydrophobic. Now, it has a hydrophilic head. I want you to remember that. Well, what makes a phospholipid different from a regular lipid? A phospholipid has a charge. It is going to have a charged head. The tail has no charge. The head has a charge. It is a hydrophilic head and a hydrophobic tail. So basically, the tail doesn't like water. The head does. The head's like, I'm thirsty. The head's drinking all the water. So it's like when you think phobic, right? Arachnophobia, or whatever, you have a fear of heights. Phobic is you hear that phobic, arachnophobia, they are basically scared of water. You can think of it like that. The tail doesn't want to get near the water, but the head is like, I don't mind. I'm hydrophilic. I can deal with it. So the hydrophilic head has a phosphate group on the inside, and that's what gives it the charge. There's no charge on the inside, so another way to think about this is the head is polar and the tail is nonpolar. So basically, like I said, the head has a charge which makes it polar, the tail doesn't, so it's nonpolar. So the inside of the cellular membrane is nonpolar. It is nonpolar, and the outside is polar. So these phospholipids line up, and the surface of the cell membrane has a charge, but the moment you get below the surface, there is no charge. The inside is nonpolar. So because the inside is nonpolar, things that are polar cannot get through. Things with a charge are not getting through the cell membrane. So how do we get important things with a charge? Well, here's what can get through the cell membrane. Little tiny things, tiny particles can get through the cell membrane, tiny molecules can get through the cell membrane. Anything with no charge can get through. But when it comes to those bigger things with a charge, we need them, they're vital. So how do they get through? Well, luckily we have these things called protein channels, and we're going to get to those in a moment, and those are the reasons why larger molecules are able to pass through the non-polar inside of the cell membrane. So the next thing we have are protein channels in the cell membrane. There are many different types of proteins in the cell membrane, but right now we're going to be focusing on the protein channels. Now the protein channels allow for things that are just too big to pass through the phospholipid bilayer. So these protein channels move larger molecules, and they move particles through facilitated diffusion, and we're going to talk about that within the next couple videos. But right now, a perfect example is H2O. And now, how does water move across the membrane? Well, it's small, yes, but remember, it also has a charge. The inside of the cell membrane is nonpolar, so things that are polar cannot come through. So luckily, there's something called aquaporin, and aquaporin is a protein that allows the H2O to move through the cells. And because of aquaporin, it controls the amount of water that's moving through. So it's moving through a tight little bind in the aquaporin. Let's throw up an image of aquaporin, please. Thank you. And now these proteins, also these channel proteins, do active transport. There's the famous sodium-potassium pump. When we cover diffusion and active transport, we are going to discuss the famous sodium-potassium pump. Every time we think of active transport, the sodium-potassium pump is the perfect example. We're caching an ATP to move sodium to the outside and potassium to the inside of the cell. Let's throw up a picture of the sodium-potassium pump, please. So they're allowing big things with a charge to just move across the membrane. That's what those channels are for. The next part is we're going to be talking about the cell wall. Let's get a picture on the cell wall. A lot of pictures going up right now. So the cell wall is similar to the cell membrane. It's filtering stuff that's coming in, that's coming out, but it gives the cell a rigid structure. So it's actually made up of cellulose, and that's why it's hard to, eat a tree. You don't eat a tree. It's difficult to eat a tree. So due to that structural integrity, bacteria, which have cell walls, are able to retain water more than a human cell, because a human cell, it doesn't have a cell wall. It can't hold on to that water. So cell membrane taking in water, a human cell will explode when there's too much water, unlike a cell wall, which can hold on to it. We'll be doing in-depth videos on active transport, passive transport, hypertonic, hypotonic, and isotonic, all coming soon. Like, subscribe before I disappear.